In the second half of chapter 3, we're going to talk about how particles make their way across the membranes of cells. So you will recall that the membrane of cells is composed of a type of membrane referred to as a plasma membrane. The plasma membrane consists of two lipid bilayers that are each composed primarily of phospholipids. And so interestingly, when we think about transport across these membranes, we have to remember that this is an extremely important aspect of our physiology. And so, for instance, in the case of nutrients that we talked about in the first half of chapter 3, nutrients are absorbed via the digestive tract, make their way into the blood, and those nutrient particles have to continually diffuse, or at least make their way, we'll talk about what diffusion is, but make their way from the blood into the cells. And as they do so, they have to pass across the plasma membrane. And so the purpose of our discussion here will be to discuss several different mechanisms that allow nutrients to get, and other things to make their way across the plasma membrane. So first off, the first way that we're going to talk about that allows substances to move across the plasma membrane is diffusion. Diffusion is very simply the movement of small particles from an area of high concentration to low concentration. Diffusion can be very conveniently illustrated by imagining a glass of water, and into that glass of water you place a single drop of food coloring. The food coloring hits the water, and the molecules of that food coloring start to spread out. So you start with a little green dot, and that green dot very rapidly spreads out until the entire glass of water has a light green tint to it. And so that's diffusion. Diffusion is very important in your body, but only certain things can cross the plasma membrane that we mentioned a moment ago via diffusion. So on this particular slide, we see the plasma membrane. We see the heads, the phosphate heads of the phospholipids that I'm pointing to with the cursor now. And of course, the phosphate heads point towards both the outside of the cell, where I'm pointing to now, and the inside of the cell, where I'm pointing to here. So this would be outside the cell up here, and then this would be inside the cell down there. The inside of that plasma membrane contains the tails of the phospholipid, the fatty acid tails. And so these are water-hating, hydrophobic portions of the molecule. And so the inside of that plasma membrane is a water-hating region. And so interestingly, when we think about things that can and cannot cross the plasma membrane, we have to think about two different criteria. One is, we have to ask, is the molecule hydrophobic, water-hating, or hydrophilic, water-loving? And if a molecule is hydrophobic, then there's a good bet that it will be able to cross that plasma membrane. CO2, for instance, is a very small hydrophobic molecule that can pass across that plasma membrane without any trouble whatsoever. Steroids are another example of a very small lipid that is very hydrophobic, very water-hating, and can cross that plasma membrane. Now this should make sense to you if you think about it, because the interior of that plasma membrane is a water-hating region, and therefore molecules that are water-hating can make their way into that region and pass through it without any trouble whatsoever. The other thing we have to ask ourselves when we think about what can and cannot diffuse through the membrane is, how big is the molecule? As it turns out, very large molecules, even if they are hydrophobic, are very likely not going to be able to cross through that plasma membrane. It's very much more likely that very small molecules can cross the plasma membrane. In fact, although this seems somewhat confusing, water itself can to some extent diffuse through that plasma membrane. There are special proteins that we'll talk about later in our class that allow water to move through the membrane in some cases. But water and other very small molecules, even if they are somewhat hydrophilic, water-loving, can pass through the membrane. So, in other words, to summarize, if a molecule is small and hydrophobic, then it can pass through the membrane very easily. If it's large and hydrophilic, water-loving, then it's not going to be able to pass through that membrane at all. So diffusion is a type of passive transport. This means that no energy is required for diffusion to occur. No pumping of any type is occurring. The molecules are simply moving because of random motion from an area of high concentration to an area of low concentration. Now, what we just discussed then on the previous slide is something that we refer to as simple diffusion. So now add that at this point to your definition of diffusion, of at least that type of diffusion. Simple diffusion is simply the movement of molecules across a plasma membrane without any sort of assistance. We want to now draw a distinction between simple diffusion and facilitated diffusion. Facilitated diffusion occurs when we have a molecule that is large and or hydrophilic and therefore cannot cross that plasma membrane. And so it still needs to cross the plasma membrane, and it still has a concentration gradient that would encourage it to cross the plasma membrane, but it can't because it can't go through the membrane. So in this case, what we have are these special molecules referred to as transport proteins. And so if we look at our screen, we see the plasma membrane, and we see 3 green transport proteins that are found in this particular membrane. And so the transport proteins very simply make a tunnel or a hole through that plasma membrane that allows a particular molecule to go from an area of high concentration to low concentration. Remember, diffusion only occurs what we refer to as down a concentration gradient. And so down would be going from an area of high concentration to low concentration. As you look at this slide, I encourage you to remember that the fact that the high concentration is on the top and the low concentration is on the bottom is of no consequence whatsoever. The important thing, it could very well be, just as well be reversed, but the important thing is that these molecules are moving from an area of high concentration to low concentration, therefore down a concentration gradient. And in this case, we have glucose, amino acids, and transport proteins as we go from left to right on our screen. Calcium, glucose, amino acids, and calcium as we go from left to right on our screen. And these are part of molecules or ions that are either large or hydrophilic and therefore cannot pass the membrane, pass through the membrane on their own. And so each one has an individual transport protein. It's also interesting to notice that transport proteins are very specific, which is not portrayed very well in our diagram here. But what you need to remember is that the transport protein for glucose, shown on the left, is not going to work for the one, not going to work for calcium. The one for calcium is shown on the right there. So interestingly, each transport protein is specific to one particular molecule and or ion. So again, this is also a form of passive transport. No energy is required for this process to occur. Alright, and so now we get to one of the most exciting parts of this second portion of our chapter, and one of the most important topics in all of biology, and that is the topic of osmosis. And so osmosis is a type of diffusion, a special type of diffusion. And so I have the definition for osmosis on this particular slide. And so we can read that osmosis is the movement of water across a semi-permeable membrane from an area of high water concentration to low water concentration. And so it's important to keep in mind then that this is the movement of water. Oftentimes osmosis is defined as the diffusion of water. And so let's take a look at the diagram on the screen, and I'll use the cursor to point, and let's talk about what osmosis is. So let's look at the left-hand portion of our diagram. This is a beaker, obviously, and in the middle of the beaker we have a semi-permeable membrane that I'm pointing to here. Now you cannot see the holes in it, but you need to believe me when I tell you that this membrane is permeable to water. Water can go through, but the solute particles, which in this case is sugar, you see all the little dots here? Those little dots, as the diagram indicates, represent sugar, not water. And so the water can go through, but the solute particles cannot. And so look at the two halves of this beaker and ask yourself this question: on which side is water more concentrated? On which side are the water molecules more crowded? And that will allow you to predict where the water will move. Because if you think back to diffusion, you remember that diffusion is the movement of particles from an area of high concentration to low concentration. Well, osmosis is a diffusion of water. And so osmosis is the movement of water from an area of high concentration, that is high water concentration, remember that, to low water concentration. So if you look at each side of this beaker then, look at the left and look at the right, ask yourself, where are there more sugar molecules? And the answer, of course, is on the left. There are fewer sugar molecules on the right. And so in this case, that means that water is more concentrated on the right-hand side of this membrane, as the diagram indicates. because on the left-hand side, the sugar molecules are taking up space, which means that there's less water. Water is less concentrated there. And so that concept is perhaps the most important in understanding osmosis. Osmosis is the movement of water from an area of high water concentration to low water concentration. And so, if you think about it this way, that's always going to be that always is going to mean that water is going to move from an area of low solute concentration to high solute concentration. And that's what happens in this case. You can see that water is more concentrated on the right. Water wants to move to an area of lower water concentration, and so if we now shift to the right hand side of our diagram, we see that water is going to move across that membrane, and it's going to move into the area where water is less crowded, and that means that water is going to be pushed up on the left hand side of that beaker. And so that is osmosis, and osmosis is very important in the physiology of your body, as we'll see over the next couple slides. Alright, now here's how osmosis affects both plant and animal cells. If we start at the top, we have osmosis in an animal cell. And so this could be a human cell or a cell from any other type of animal. If we place that cell into a solution that has a higher solute concentration outside the cell, then we have to ask ourselves, where is there more water? Is there more water inside the cell or outside? And the answer, if the solute concentration is higher outside, is that there's less water outside and more water inside. That means water is going to go from inside the cell to outside the cell, and the cell is going to shrivel as a result of that exodus of water. And so this is important, and we'll talk about how it influences the solute concentration in your cells and in your blood in just a moment. Let's first take a look at what effect osmosis has on plant cells. Now, plant cells have a very distinct difference than animal cells. Plant cells have a cell wall that animal cells do not have. And so, in the second half of our diagram there, we see that if we take a plant cell and we place it into a solution that has a higher solute concentration outside than inside, then water leaves. But in this case, the cell wall can't move. The cell wall is completely rigid. But something referred to as plasmolysis occurs. Plasmolysis occurs, as we can see in the lower right-hand quadrant of our diagram, when the plasma membrane pulls away from that cell wall and shrivels up inside the cell wall. And so, interestingly, even though the plant cell can't collapse, the plant cell can shift just a little bit and become a little bit flaccid. And so you're familiar undoubtedly with what happens when you don't water plants. And so when plants are not watered, then they become limp and they wilt. Interestingly, it's the water inside the plant cell, and so the plant cell has to be in a healthy, non-wilted plant, like we see on the bottom left-hand side of our diagram. There's water in the plasma membrane, and water is pushing out against that cell wall, making that cell rigid. And when thousands of those rigid plant cells are placed end to end, then it allows a leaf to remain upright in the manner that it needs to in order to collect sunlight. So osmosis is extremely important in plant cells as well. And plant cells prefer to be, and typically are, in an environment. They have to be in an environment in which there is a greater solute concentration inside the cell than outside the cell. So interestingly, that's why plants that are exposed to salt water have an extremely difficult issue, as you can see. Things like mangrove trees, for instance, that grow in the ocean. If you go to Tampa, actually let's go a little bit further south. If you go to Naples, Florida, and go to the ocean there, you'll find vast mangrove forest just off the coast of Naples there. And those are trees, a type of plant, that are submerged in salt water part of the day. And so they have a very difficult, they face a very difficult issue because there's a very high concentration of salt in salt water, about 3.5%, and that's higher than what you'd find inside a normal plant cell. And so interestingly, they deal with this by maintaining a very high concentration of solutes inside their cells. If you compare a mangrove tree root to an oak tree root, they'll have a much higher solute concentration inside that mangrove tree root versus an oak tree. Okay, let's move on. Now, to briefly review osmosis then, let's take the opportunity of this slide to do that. So first, let's look at part A of this diagram. And in this case, we have a bag that contains 2% sucrose. And so this particular bag is a semi-permeable membrane. In other words, water can get across, but sucrose cannot get across. And so first we take that bag and we place it into a beaker that contains 2% sucrose. And the question then is, what happens to water here? And interestingly, in this case, the solution is what we call isotonic, a solution term rather that you can find under B on the right-hand side of this diagram. Isotonic means that the solute concentration, not the water, but the solute concentration is the same on each side of the membrane. And so in this case, water does move. Some water moves out, some water moves in, but there's no net movement of water. Overall, the volume of the bag stays the same. Now if we take that same bag, and so we're still on part A of the diagram here, and we put it into the 10% sucrose, what we see is that the bag loses volume in that 10% sucrose, because we realize that there is 2% sucrose in the bag, 10% sucrose outside the bag, and this means that there is more water inside the bag than outside, so water is going to leave the bag. And so in this case, we are placing the bag into a hypertonic solution. And you can see that term under C on the right-hand side of your diagram. If a cell, or in this case a bag that's mimicking a cell, is placed into a hypertonic solution, it will shrivel up and shrink. The last question then is, what happens if we take the 2% sucrose bag and place it into pure water? In this case, we're taking the bag and we're placing it into a hypotonic solution. And remember these terms, and you can find hypotonic on the right-hand side under part D of that diagram. These terms refer to solute concentration, not water. And so the way we use these terms is as follows. In this last example, when we take the 2% sucrose and put it into water, we're now on part A on the right-hand side of the diagram. Then we say that the 2% sucrose, that the inside of the bag is in a hypotonic solution. The inside of the bag is hypertonic, the outside of the bag is hypotonic, because the outside of the bag has a lower water concentration. And so what happens in that case? And the answer is something you can figure out on your own. So first ask yourself, where is there more water? Is there more water inside the bag, or is there more water outside the bag? And in this case, the obvious answer is there's more water outside the bag. There's absolutely no solute, so there's pure water outside the bag. So where does water want to go? Water wants to go towards the lower water concentration, which means the higher solute concentration. Water goes into the bag and the bag swells. Interestingly, what we saw in part A there is a direct replica of what would happen if you took your red blood cells, the cells of your body, and placed them into an isotonic, a hypertonic, and a hypotonic solution. In part B of our diagram, we've shifted over to the right-hand side of the diagram now, we see red blood cells as they should appear, as a biconcave disc, and in this case they are immersed in an isotonic solution. The solute concentration outside is the same as inside. And so as you would guess, your plasma, it provides an isotonic solution for your red blood cells. Your blood is nothing more than, little more, let's say, than cells, red blood cells, along with some white blood cells that are floating in plasma. And the plasma can't be just pure water. Plasma is mostly water, but it can't be pure water, because if it was, what would happen? And you realize that it would be like the situation that we saw in part A on the far right hand side there. Your red blood cells would swell and burst. But so your plasma provides an isotonic solution for your red blood cells. If we look at C, part C, then we see red blood cells placed into a hypertonic solution. And in this case, it's just like the middle beaker on the left-hand side of the diagram. Your red blood cells are shriveling because water is moving out of the red blood cells. In part D, we have placed red blood cells into a hypotonic solution. And in this case, the red blood cells are swelling, and they may eventually burst, depending on the difference in concentration inside and outside the cell. But in pure water, they very well may burst because water is moving to that lower area of water concentration inside the cell. So osmosis is very important. I encourage you to look over these slides again, make sure you understand it, read about it in your text, and before you take the quiz on osmosis. Finally, let's move on to our next type of transport, and our next and last type of transport will be active transport. So active transport is different than the two types that we discussed previously, and I'm making two categories of what we've talked about previously. We talked about diffusion, both simple and facilitated, and then we talked about osmosis. Both of those are passive types of transport. That means they do not require energy. Here we have active transport. Active transport does require energy in the form of adenosine triphosphate, or ATP. We'll talk more about ATP in Chapter 4. But for now, we simply need to know that ATP is the energy currency of cells. Whenever the cell wants to do anything, it has to have ATP to accomplish that. Just like you have to have gas to make your car go, or electricity to turn on the lights, well, your cell has to have ATP to make anything happen inside the cell. So, in the case of active transport then, the key is this, and I will ask you this on the quizzes and exams. How do you recognize active transport? There's a couple ways to do it, but one thing that's a dead giveaway is that the molecules are going to move from an area of low concentration to an area of high concentration. In active transport, molecules are going to be pumped up a concentration gradient, up a concentration gradient. They're going to go from an area of low concentration to an area of high concentration, as is depicted on our slide here. If they were moving from an area of high concentration to low concentration, then it wouldn't matter. You wouldn't need active transport. You would just have diffusion. But they're always going to be going from an area of low to high concentration in the case of active transport. Active transport requires a transporter protein that we see depicted on the screen here, but the difference in this case is that the transporter protein is an actual pump. And I mean the difference between this and the transporter proteins we saw in facilitated diffusion. In this case, the transporter protein is actively pumping these ions across the membrane. An interesting example of how active transport is used in your body is in the nervous system. We'll talk a bit more about the nervous system later, but it's fascinating to realize that every time you move a muscle or experience a sensation, like tell if something is hot or cold, or actually have a thought, that's all the result of ions that are moving back and forth across nerve cells in your body. I mean, it's a fascinating idea, and so you may realize that something like Novocaine, for instance, which is a slightly outdated type of anesthesia used in dentistry, they use a later version of it now, is a drug that simply blocks the movement of ions across those membranes that we see on the screen. And so fascinatingly, in order for your body to function, active transport has to continually be working. Your nervous system entirely relies on active transport, as we'll cover a bit later in our class. Another example of active transport, the importance of active transport in your body, is the movement of glucose into certain cells in your body. Glucose moves into some cells passively, but other cells require an active transport of glucose in order to function. So remember, glucose requires energy in the form of ATP, and I'm sorry, I think I said that incorrectly. Remember that active transport requires energy in the form of ATP, and remember that active transport requires that molecules move from an area of low concentration to high concentration. Finally, we're going to finish our discussion of transport across membranes by talking about something called bulk transport. What happens if you need to transport a large number of molecules, or something quite large, say a cell across a membrane, a smaller cell across a membrane? And so in this case, we use primarily two types of bulk transport. One is exocytosis. And so both of these are shown on our slide, in the diagram on the lower portion of the slide. Exocytosis occurs when, if we start here, I'm going to point with the cursor here, if we start here with this vesicle, a topic that we'll cover a bit later in our class, then the vesicle is simply a ball of plasma membrane. It's surrounded by plasma membrane, and that vesicle goes and it merges with the plasma membrane, and it dumps the contents of that vesicle outside the cell. Interestingly, when this vesicle interacts with the plasma membrane, it's much like when two soap bubbles interact. When you were a kid, you blew bubbles, and you recall that two of those bubbles could run into each other, and they'd stick together and form this little partition between two bubbles. And then that partition could pop, forming a larger bubble. And that's much what happens, very similar to what happens here in the case of the interaction between the plasma membrane and the vesicle. This vesicle runs into the plasma membrane, that surrounds the cell in the case of exocytosis, and it becomes one with that plasma membrane, dumping the contents of the vesicle outside the cell. Endocytosis is not hard to imagine if you understand this, but it's simply the opposite. And so in endocytosis, a large indentation forms in the cell, and we can see this on the sort of right-hand side of this diagram that I'm pointing to now. So here's a food particle, an indentation forms in the plasma membrane around it, and then that indentation breaks off and goes into the interior of the cell, becoming a vesicle. And so in this case, we're showing a food vesicle, and so another vesicle in the cell called a lysosome fuses with that food vesicle. The food is digested, and then, as we pointed out previously, the waste particles are gotten rid of via exocytosis. It's also interesting to note that things like white blood cells use both endo and exocytosis. Some types of white blood cells actively seek out bacteria at the site of an infection. In your body, they do this, of course, and they engulf those bacteria, and they destroy the bacteria, and then they get rid of the parts of the bacteria via exocytosis. So they use endocytosis to engulf the bacteria and exocytosis to get rid of the waste products that are left after they have killed and digested the bacteria. This concludes our discussion then of transport across membranes, which brings us to the end of Chapter 3